Looks like we got a good group of people here tonight. I'm going to just provide the introduction for tonight. My name is Tom Hodgman. I'm a Katahdin breeder from northern New England, and I've been in NSIP for 15 years now, so I'm pretty familiar with it. I'm also serve on as the Katahdin breed representative to the board of directors for NSIP. Lately, we've been doing a lot of work with the Katahdin Hair Sheep International Association in the NSIP committee to develop an effort towards commercializing genomic enhanced breeding values for the U.S. sheep industry. We're starting off with Katahdins because we have we now have the genomic reference population available to initiate that effort, thanks to work by Dr. Joan Burke and her colleagues. Part of this commercialization effort, that's really a two-pronged approach. The first is to educate folks as to what genomics are and how it can be used in your flock to improve your selection efforts, build better sheep, and we're doing that through a combination of written documents, sort of the old school approach, and there's a number of us working on those right now as we begin to nail down more and more about the how this is. I see. And as well as a webinar series, and you're here at the first of the 4 webinars that were set up by Dr. Andrew Weaver. Andrew is not available to be here at this moment because he had some travel delays today with some flight delays. So he will be joining us, but he was unable to introduce the presentation tonight. As you learn tonight, and in the next 3 webinars, excuse me, we really have a new tool available to us. It's right on the horizon for a number of sheep breeds. The Katahdin breed will benefit from it first off, but the other breeds, with a few things in place, will be able to take advantage of that as well. This is really a new tool for selecting superior breeding stock based on not only pedigree, pedigree relationships from genetics, but actually a more thorough view of the animal from a genomic standpoint. And I'll let Rusty explain more about that. But this is quite new to the sheep industry as a whole in the U.S. It's been commonplace in other livestock breeds in the U.S. for a number of years, but this is really the first time we've had this opportunity in the U.S. sheep industry. It's common in the sheep industry in Australia and other places, but not here. So I'm really excited to introduce our speaker tonight of webinar number 1 on a new age in sheep breeding, and that's Rusty Burgett. And Rusty is our coordinator and director of the National Sheep Improvement Program, the national program for genetic improvement in sheep in the U.S. Rusty did his undergraduate work at Ohio State, and then on to University of Wisconsin. He worked in nutrition, and then subsequently worked with the Spooner flock of I believe it was a dairy sheep flock, and then about 6 years ago came to NSIP to help us as the program coordinator. He's really been the person behind making the genetic evaluation run in the U.S. A lot of behind the scenes work, and there's really nobody else out there that's able to step down this complicated topic and with such a thorough understanding of it down to the individual producer level. And I really appreciate his advice to me and to other sheep producers. So Rusty has played a really active role in helping us launch the effort at genomic enhanced breed values for all sheep producers in the U.S. And I want to thank him for that effort and turn it over to him. You good to go, Rusty?
I'm good to go. Thanks for that, Tom. And thanks to everyone for joining us here this evening. Hopefully it's warmer wherever you all might be. We were at 24 below and it's not letting up. So I hope everyone is nice and warm and cozy here this evening for our presentation. So, as Tom talked about, my role here tonight is I wanted to kind of give you from the producer's perspective, some of these new technologies that are coming down the line. We've got some great speakers on the agenda here for the next couple webinars. So they can talk to you about the technical capabilities of these new tools. I'm going to give you kind of the 30,000-foot view in simple sheep herder terms, because me trained as a nutritionist, that's my understanding level, and I really lean on those technical experts you'll hear from in the next few webinars on the technical details. So before we get into that, just need to give the quick acknowledgements to Katahdin Hair Sheep International, who's really spearheading this effort for this webinar series, and we'll get into the details of the project later on. But this is funded by a project through the USDA National Sheep Industry Improvement Center, as well as NSIP and several of our land-grant universities. So a lot of different hands here at work to make this effort possible. And if anyone has any questions, feel free to use the chat function. I'll try to keep an eye on that as we go along. If you need to ask a question, feel free to go ahead and unmute yourself and stop me and ask that question. I would ask, though, if you're not speaking, if you could please mute yourself, just so we can avoid any feedback and echoes and that sort of thing. All right, so we're going to go ahead and get into the details here. What are we talking about? Well, I think since we're talking about new kind of cutting edge technologies, that we should take a step back and look at where we've been. And I think it's always good to kind of get back to the basics as to what we're trying to do with these genetic selection techniques. I mean, in essence, in genetic selection, all we're trying to do is we're trying to match up ewes or females with the group of males to produce offspring. Pretty simple on its face. Pretty simple techniques here to get us started off with the very basics. It gets a little more complicated, though, when we're looking at, I guess, what are we actually trying to do in terms of this next generation? Really, what we're trying to do is we're trying to figure out which genes these females and these males are going to pass along to that next generation. Okay, really, it's those genes that we care about in making our breeding selection decisions. Okay? Because as we'll get into in here in just a little bit, it says those genetics that kind of provide the blueprint or the framework that we're going to build a productive, profitable animal upon. Okay? And in today's modern agriculture, it really requires us to produce, as producers, to utilize as much information as we possibly can, to be as efficient as we possibly can. It's just the modern world that we live in. We have to make sure that we are efficient at every step of the production chain, and that really starts with genetics. Okay? So, how do we make our genetic selection decisions? Well, one thing that we've been doing, we've been selecting sheep for 10,000 years this way through visual appraisal. We have to look at these animals. We have to use what we can see in front of us to try to make an educated decision on whether this animal is going to produce a productive, profitable offspring for us. Okay? The trouble with that is that we know that looking at visual appraisal of an animal, we know that these animals are both a combination of their genetics that they inherit and the environment that they are raised in. Okay? On average, about 80% of what we see in an animal is actually due to the environment that the animal is raised in. But when we're interested in this animal as a breeding animal, they don't pass along that environment. They pass along those genes. Okay, that's what we need to try to figure out. We need to try to quantify that genetic portion that's actually going to make our next crop of ewes or lambs for our production systems. So that's where it gets a little bit tricky. We need to find some tools that'll help us to shed more information on what is that genetic portion. what genes are actually going to be passed down to the next generation. Okay, because again, every animal is a combination of both its genetics and its environment. Okay, it's this genetic portion that gets passed from generation to generation, and it's up to us as shepherds to provide these animals with the environment to express those genetics. Okay, but again, this environmental portion, it doesn't get passed down. It's the genetic portion that does. Okay, and that genetic portion, we'll start off being technical, and then we'll simplify from here. Okay? This actual genetic portion that's passed down from generation to generation, okay, that comes in the form of these chromosomes, which are made up of DNA. And this DNA is essentially a blueprint. Okay, it's a blueprint that tells this animal how to be put together and how it's supposed to perform. Okay? Then it's up to us as producers to provide the building blocks, provide the materials for those animals to fulfill that blueprint and be productive for us. Okay, but it's this DNA, this blueprint here, that determines how that animal is going to perform for us. And that DNA, okay, what they actually inherit from their sire and their dam, that's what we call the true breeding value of an animal. That's how those strands of DNA or how those genes come together. That's the true genetic potential of that animal. Okay, and in our genetic selection exercises, what we're trying to do is we're trying to estimate that true breeding value or that true genetic potential. Okay, so everything's going to come back to this, trying to use as much information as we have to try to figure out what is exactly that true breeding value of the animal. Okay, so looking at this ram right here, okay, we can't really see from the outside what that true breeding value is or what that blueprint says this animal is supposed to perform like. Okay, so we need to add some extra information, and I've got an analogy that I'd like to use here throughout the evening. Might be a little bit off from what you're used to hearing, but in my mind it makes sense, and I think a new way to look at it is always beneficial. So my analogy here, okay, this is the wall behind me. This is the wall in my office. And a neighbor of mine just the other day, he dropped off a sign that I wanted to hang up on this wall. Okay, so I want to hang that sign up right here in the middle of this wall. So I'm going to take a hammer and a nail. I'm going to pound that nail into this wall so I can hang that sign. But it's pretty heavy, so I want to make sure that that nail hits a stud in that wall. I want to hit that 2x4 stud or the framework so it's nice and well supported. Okay, so if I'm going to just use visual appraisal, just like I do in selecting sheep, okay, I'm essentially just taking a random shot and just putting the nail up to the wall here and just smacking it with a hammer and hoping that it hits a stud. When we're looking at a ram just based on visual appraisal and trying to guess what his performance is going to be, okay, that's just like holding that nail up to the wall in a random spot and hoping, praying that I hit a stud. Okay, because of that heritability component, because of the majority of what we see in this ram here being controlled by the environment, okay, we don't have much information to go on to try to hit that stud in the wall or try to estimate what his genetic potential is. Okay, I think we need a little extra information, and what a lot of us are probably used to looking at is phenotypic information. Okay, phenotypes being just the information that we can collect or observe on these animals at any given time. So phenotypic information would be the fact that this ram was born a triplet and raised as a triplet, and that his birth weight was 8.9 pounds. His weaning weight was about 65 pounds. His post-weaning weight was about 140 pounds. Okay, so it's all phenotypic information, things that we can observe about this animal that helps to shed a little bit more light on his genetic potential. So I'll go back to my wall example here. Okay, rather than just taking a random shot in the dark and trying to hit a stud in the framework here, I need to apply some phenotypic information. I need to use my powers of observation to try to shed a little more light on this situation here. Okay, so that phenotypic information in this situation is, well, I see a couple seams here in the wood paneling on the wall. That might give me a little bit more information. Okay, or I can do the old tried and true method of knock on the wall and try to see if you can hear for that stud in the wall. Okay, see if you can hear the pitch change in those knocks, utilizing our powers of observation to try to find that stud. Okay, this analogy goes a little bit further because similar to visual observation of breeding sheep is that sometimes these phenotypes can be a little bit misleading. Okay, I've got a lot of building experience. I grew up building houses with my parents, and my powers of observation would tell me that if I see a seam here, chances are there's a stud behind that seam. Well, in this situation, that phenotype, what I would observe, was actually very misleading. There's actually not some framework back behind there. It's actually somewhere in this region. So utilizing phenotypic selection, I've narrowed it down that there's a stud somewhere in this general area of the wall. Okay, there's still a lot of room for error. That's about a 6-8 inch gap there, and I'm trying to hit about an 1.5 inch wide stud within that. So I've narrowed it down a little bit, but there's still a lot of room for error there. So I need to use a different tool to try to help shed a little bit more light. This information is great. It's certainly better than nothing. It sheds a little bit of light on what this animal's true breeding value is, but it doesn't get me all that close. So that's where we step into estimated breeding values through NSIP. So if you take that phenotypic information and you submit that data to NSIP and it gets run through the genetic evaluation, that phenotypic information is then going to be compared. It's going to compare this ram to all the contemporaries that this ram was raised with. It's going to compare it to the performance of his sire and his dam. It's going to compare it to all of his known relatives within that same flock, across a bunch of other different flocks. All of that information is building statistical power in that genetic evaluation, and we're getting a more precise picture about the genetic potential of this animal. Okay, so that's you can see here, these are the estimated breeding values. Hopefully everyone's familiar with estimated breeding values through NSIP. We can see here, so rather than just looking at the type of birth and type of rearing as born triplet, raised triplet, we can now see that the genetic potential is a 4.8% for number of lambs born, 21.4% for number of lambs weaned. Then we can go into our weight traits, weaning weight, post-weaning weight, eye muscle, and then his index here. Now what we can see here is, along with each of these estimated breeding value calculations, we also have an accuracy value. Okay? So the more information we have, the more accurate that estimated breeding value is going to be. Okay? And what this accuracy value actually represents is how much or how close this estimated breeding value is to the true breeding value of that animal. Okay, so this one right here is a 2.0 and a 96% accuracy. That means that we have tons of information going into this EBV calculation, that we're 96% accurate that this is getting close to the true breeding value of that animal. That's what we're trying to do, is we're trying to get estimate that true breeding value of that animal. Okay, so we'll go back to our wall example here. Okay, we just used the phenotypic selection. We did a knock and we figured out that our stud is somewhere within this wall. Now I'm going to use some technology similar to estimated breeding values. I'm going to use a digital stud finder. Okay, so utilizing that technology, using that stud finder, I've then narrowed it down to about a 3-inch range here on where that stud might be. I went from a 6 or an 8-inch range, narrowed it down to about a 3-inch range, okay, which is, I'm getting a heck of a lot closer. It's a very powerful tool using this stud finder, using these estimated breeding values, but it's still not 100% accurate. Okay, it's still not getting to that precise level that I'm really wanting to. Okay, and a lot of that comes down to the amount of information we have on these animals. Okay, this ram right here, he was born in 2010, and he was a very widely used stud ram in the polypay world. A lot of my ewes and my personal flock go back to this ram right here. Okay, he actually has over 400 progeny recorded in 4 different NSIP flocks. Okay, so it's that vast amount of data that is leading to these high accuracy estimated breeding values. Okay, it's getting us a very accurate depiction of his true breeding value. But what if we look at this this ram lamb here that I have used pretty heavily this year? This is a fall-born ram lamb, so he's just a yearling right now. He has 0 progeny. Okay, so he has limited information going into the database. And you can see that reflected in these estimated breeding value accuracies over here. Okay, you can see that his reproductive estimated breeding values are in the 40% range. Okay, his growth EBVs here are in 65, 67% range. Eye muscle depth is even further down. Okay, so for these young animals with limited information, we need an even better tool, a more precise tool to help us out with some of these decisions. Okay, go back to my example here. Okay, most of you probably never thought that I could milk this much information out of a simple photo of a wall, but we're going to milk this one for all it's worth. Okay, so we used our stud finder to narrow it down, but now we're going to use a more precise tool. If I actually had a blueprint of my house, okay, I could go to the actual blueprint, okay, and figure out exactly where those studs are within that wall. Okay, so it's getting even more precise, getting even more basic on the scientific level. I know exactly what the engineers drew up for where those studs are supposed to be placed in that wall. I can then use this information to precisely put my nail in to the wall where I absolutely need it. That's where these genomic evaluations are coming into the sheep world. Okay, it's getting more into that basic level. It's looking more at the actual blueprint of those animals to see what genes they physically inherited from their sires and from their dams. So we're looking at the actual DNA, the actual blueprint of that sheep. Okay. To get these genomic evaluations, it's a fairly simple process for sheep producers because really it simply starts off with sending in a DNA sample for evaluation. So what we've kind of settled with in our current evaluations is using these right here. If you're not familiar with these are called tissue sampling units, or TSUs. Okay, and this is very similar to an ear tag gun here. These are individual TSUs, and what these do is these take a very small hole punch out of the ear of that lamb. Okay, so it simply takes it's about the size of a pencil eraser. Takes a little hole punch. Okay, and it takes from this gun, it drives that little ear notch into a little vial of preservative down here within this, and we have an individual DNA sample on an individual lamb. Those DNA samples are then sent in to a genotyping lab, okay, where the DNA is extracted, and they're running through different genotyping platforms. They're figuring out differences in single nucleotides. Okay, so when these DNA come together, they're looking at differences in these combinations of DNA at different locations across different animals. And then what we need to do at NSIP is try to correlate these differences in DNA to differences in performance. Okay? So I'm sure Dr. Lewis will shed a little bit more light on what these single nucleotide polymorphisms are, these SNPs. Okay? But it's simply, we're looking at differences in the DNA and correlating those to differences in production. It's a pretty in-depth process. I'm not going to try to shed too much light on that. I'm really going to lean to Dr. Lewis to do that. But it's so complex because I'm sure most of us know that there's 54 chromosomes within a sheep, but those chromosomes are made up of billions of base pairs within that DNA. Okay, so if we look at these chromosomes, again, they're made up of DNA. There's billions of base pairs, so billions of places where these DNA can differ. What we're kind of looking at is that we know that there's certain sections of this DNA that code for certain things within that animal. Now, these certain sections that are common across animals that code for certain things across these animals, those are called genes. Okay, so we're looking across the entire genome of the sheep through these genome-wide association studies, these GWAS studies, to find areas across those billions of base pairs of interest. They've done those studies, and they've then taken those areas of interest and they've put them on these things called SNP chips. Okay, there's low density chips, medium density chips, and high density chips. The density just refers to how many locations across that genome these chips are looking at. Okay? So low density, they're looking at 15,000 different locations across that genome. The medium density chip, which NSIP is using, is looking at 50,000 different locations across that genome. There's high density ones that are looking at 600,000-700,000 different locations. Those are mostly used for research purposes, but we're going to take these 50,000 different locations and apply it to our genomic evaluation. But because this thing is so huge, because there's these billions of base pairs that we're trying to look at, sometimes we might not hit the nail exactly on the head. We might not be able to narrow it down exactly which areas of those DNA are coding for certain things. So one more trip back to my office wall. Last little bit of this analogy I'm going to subject you to. Sometimes we're not going to get a complete DNA picture. We could sequence the entire genome of the sheep, but it's going to be extremely expensive for producers to do. So there's just, it's not economically viable. But what these tools allow us to do is utilize the information we do have to estimate the areas of interest. So again, in this example, I'm interested in a stud in this wall in the middle location right here. I don't have a complete blueprint, but I do have a blueprint of the wall over here. Now I can use the fact that these studs are 16 inches on center, and I can use the information that I do have to shed information or shed light on the area of interest that I particularly have. That's a lot of what these 50K chips are doing. So we might not be looking at exactly the right location for a particular trait, or a particular trait might be controlled by several hundred different locations across that genome. What we can do is we can utilize the locations that we do know, the information that we do have, to give us answers for those areas that we are particularly interested in. So these genomic evaluations, what are they going to mean to us as sheep producers? Okay. Well, there's three main benefits that we're going to get out of these as we incorporate this information into the evaluation. First and foremost is it's going to allow us to get an accurate parentage evaluation on our animals. The benefits of this cannot be understated. This is, if you've been in the sheep industry for any amount of time, you've probably all experienced a ram that jumps out, or more than likely 2, 3, 4 rams that jump out at a time that they're not supposed to, breed ewes that they're not supposed to, or you have breeding groups that get mixed up, and you're trying to play the guessing game of who sired which lambs. Situation that I run into quite frequently, more often than I would like to admit, is when you walk out into the lambing shed, you have 3 ewes that have lambed, a pile of 7 lambs in the middle of all 3 of them, and you're trying to figure out who goes to who. Okay. Utilizing these DNA tools, we can know with great accuracy who the true parents or pedigree animals are within our lamb crops. Okay, and that's going to allow us to define genetic relationships more accurately. And this defining genetic relationships, that will help us to find maybe in the current evaluation we have animals that really are related, but if we have incomplete pedigrees, the analysis isn't going to know that just yet. Or it might correct some things. We might have a pedigree wrong, and we might think 2 animals are related, and they're not. So even in the current quantitative genetic evaluation, just simply having accurate pedigrees, accurate parentage evaluation will propel our accuracies by leaps and bounds. Just to give you a little bit of a reference, in the dairy industry, when they started to use widespread adoption of genomics, they actually found an 18% error rate in their pedigrees. Okay? And that to me is mind-blowing. It's an industry that is almost 100% artificially inseminated. So you know exactly which straw of semen you're putting into which cows, and they found an 18% error rate in their pedigrees. So simply getting accurate pedigrees is going to be great for our evaluations. Secondly, is we're going to be able to kind of kill 2 birds with 1 stone, and we'll also have a good evaluation of our known genetic conditions. Okay? A lot of us are probably already sending in samples for things like scrapie codon testing, or TMIN 154, OPP resistance, or myostatin. Okay? These 50K chips, we can do all of this in one test, all at the same time. So you can get those known genetic conditions. And then on top of that, we're also going to be building accuracy into our genetic evaluation. Okay? So by doing this, by having these accurate parentage, and by also having more DNA information on specific locations across the genome that are affecting our performance traits, we can build accuracy within our genetic evaluation. Now, it's important to note here that it's not going to be wholesale boosts in accuracy across the entire slate of estimated breeding values. Okay. Where we're going to see the most benefit to these tools is building accuracy in our evaluations for difficult or expensive traits and lowly heritable traits. So to expand on that just a little bit, these difficult or expensive to measure traits, things like disease resistance, feed efficiency, seasonality, meat quality. These are things that are more difficult for us to, as producers, to measure on our farms. So hopefully we can utilize research populations and then utilize the genomic information from those research populations to generate estimated breeding values, genome enhanced estimated breeding values for our commercial sheep producers. Other advantages we're going to see are going to be in our sex limited traits, things like litter size, those traits that are only expressed in one sex. That's where we're going to see another bang for our buck. We're also going to see a great benefit in those traits that are measured later on in the animal's life, things like longevity or stayability. Okay, we're going to I'll show you some information in one of our colleague industries on this right here, and the ability these genomic tools can have to help out with our evaluations. Just as importantly as this increase in accuracy and the benefit that producers get out of these genomic tools, it's also important to realize what genomics will not do. Okay? Genomics will not replace the need to be collecting data on our farms and ranches. Okay? If anything, we're actually going to need to be collecting even more data because we know that the more data that we can send into these systems, the more of these SNPs we can correlate with performance data, the more accurate these tools are going to be. This is one of the most common myths that I hear as I travel the country talking to folks, is that a lot of folks think that once we get these genomic evaluations, all you have to do is collect DNA and it solves all of our world's problems. Okay? So I want to hammer that point home. These genomics will not replace the need for on-farm data collection. We're always going to need to do that. We're always going to need to have accurate pedigrees being reported on these animals as well. These tools also, they're not going to benefit the traits that we already measure or evaluate well. Think back to that old stud ram that I showed you. I mean, he had accuracies in the 90% range for most of his traits. Those traits that we can measure very well on our farms, things like body weights. Those are also typically highly heritable traits. Our quantitative genetic evaluation already does a pretty darn good job of evaluating those or getting close to that true breeding value. So these genomics aren't going to help out very much right there. Okay. And our last point here is something I'll get into a little bit later, is that these genomic tools are not going to magically appear in all of our breeds. It takes a pretty substantial research and development effort behind the scenes to get these available. As Tom alluded to, these are we're pretty darn close in the Katahdins, but I'm going to shed some more light on that, what our other breeds are going to need to do to have access to these extra tools. So now I want to shift gears just a little bit and kind of go through an example that might shed a little bit of light on the benefit of what these genomic tools can do. So we're going to go to our colleagues in the dairy industry. And with their traditional genetic evaluation prior to genomics, the problem that they had is that their main trait of interest, milk production obviously, is only expressed in one sex. Okay, it's only those cows that are producing milk for us. But what we need to try to do is evaluate the genetic potential of the bulls that are breeding those cows. So you can see where it gets a little tricky. Okay, we're trying to estimate a trait that's not expressed in the bulls that we're trying to select. Okay. Another problem they had is a very long generation interval. So what they had to do traditionally to prove a bull is they had to try to take 100 daughters from a bull to prove his EBVs. So they had to take a yearling bull, collect semen on that bull, generate 100 daughters out of that bull, and then wait for those heifers to calve for the first time, freshen in, and start producing milk. Okay, so it's 3 years until that daughter actually starts producing, because they won't breed her in the first year. She'll calve as a 2-year-old. She'll go 305 days in a lactation. And as you can see, with that timeline, that bull was already 4 to 5 years old before his daughters are in production, and we're proving his EBVs, or expected progeny differences, in beef or in dairy. Okay, so that's a very long generation interval. We're trying to shorten that generation interval so we can identify the genetic potential of these animals at a younger age. So a solution that was put together was to genotype these animals and try to identify those animals at the younger age. So they genotyped 200 cows, okay, in this particular study. From the genotypes of those 200 cows, they selected what they thought were the 100 best to produce 50 bulls out of those 100 cows. They then genotyped those 50 bulls just so they could select the top 5 bulls and then use those extensively across the registered and commercial females. Okay. By doing this, by utilizing these genomic tools, and by having an accurate estimate of the genetic potential at a young age, they reduced the generation interval from 6.2 years to 2.1 years. Okay. Now, I think it's important to realize some of the indirect costs. Not only are they getting a better evaluation of the genetic potential, they're able to figure out who those elite sires are going to be at a younger age. Economically, they're able to reduce the number of subpar animals that they have to raise out and get proved up. Okay, just think of that. If they're raising 100 bulls and trying to find the top 5, you just fed 95 bulls for 4 to 5 years trying to get them proved up. Okay. We see the same thing in our sheep industry. As we're trying to build accuracy on some of these, we keep these some yearling rams or some mature rams around, just by simply identifying which ones are worth keeping around, which ones are feeding, send the other ones down the road. We can improve our overall bottom line. Okay. So with the increased accuracy in their genetic selection tools, increased selection pressure by decreasing that generation interval, and by eliminating those animals that didn't need to be fed out to mature size, or through the genetic evaluation, they were able to increase net returns by $110 per cow by utilizing these genomic selection tools. Okay. Just imagine the capabilities that we can have in the sheep industry when we already have a much shorter generation interval. Okay, it's already much shorter than 6.2 years. If we can bring that even further back, if we can know the genetic potential of an animal shortly after birth, there's some real savings to be had there in developing these breeding animals. So how do we build these platforms in sheep? This is something that the Katahdins have already done, but this is kind of the next step for the additional breeds to have access to these genomic tools. Well, that starts off with a reference population. So these tools, they essentially need to be trained. We need to tell the computers that we need to be looking at this particular area of the genome to be getting the information we need for the genetic evaluation. Okay. So to do that, we need 3,000 animals per breed. Those 3,000 animals, they need to have pedigrees recorded on them, phenotypic measures that have been recorded and submitted to NSIP for analysis. They need to be genotyped on that 50K platform. And they also need to represent the genetic diversity of that breed. So we need to be sampling both sexes, older animals as well as young animals, and we need to represent the genetic diversity. So if we run the risk of only selecting or only genotyping a small subset of the population, we're worried that those tools won't be then applied to the overall, to the broader population. So we need to make sure that those animals are spread out geographically, spread out by management type, spread out by genetic lines, so that we can capture the genetic diversity within that breed and build that into these tools. So after we establish that 3,000 head reference population, we then need to have continued recording, data recording and genotyping of those animals. We need to keep feeding information into this evaluation because we know that the more records we have, the more accuracy we can build into these genetic evaluations. We're not going to replace the need for collecting phenotypic information. And that kind of going back to that the point of that these evaluation tools are not going to just magically appear, is that if we want to develop these novel traits, things like longevity, things like aseasonality, we need the phenotypic data sets in order to build those tools. Okay, we need the phenotypes in our research populations, and most likely in our commercial producers. We need to be collecting these phenotypes so we can keep training these tools to be the most efficient possible. So where are we currently? Tom kind of alluded to this a little bit earlier. The Katahdin reference population has been established and it has been genotyped. A lot of that effort was through a grant project that Joan Burke was heading up. A lot of different entities involved there, a lot of different researchers involved in that. But they did a lot of the background work to get that reference population genotyped. And that information has been sent into the evaluation, and we have done a trial run and received back genome-enhanced EBVs on a research basis. There's still a little bit of testing yet to be done. We just had a meeting with the folks over in Australia yesterday that our confidence level in this is just about ready to turn that switch on, but there's still a little bit of R&D we want to do to make sure these tools really are giving us the answers that we want out of them, or the answers that we were expecting to get out of them. Okay. So those will hopefully be ready to go in the very near future. Okay. But one of the other big things that we've been working on lately, this has been a massive undertaking, and this leads into the current project, the webinar we're on tonight, is that we've also developed the work plan, or the conduit to offer these genome-enhanced EBVs through NSIP. That's been a big undertaking, just the simple logistics of the several hundred producers we have spread across the US. How do we get these DNA samples collected? How do we get them organized? How do we get them sent in for evaluation, all in an organized fashion? And then utilizing that DNA as an actual reference or a resource. Okay, so these other breeds, okay, this is, I said the Katahdins, we've had this process already established. All the other breeds in the United States, we still need to build or establish and genotype that reference population. We still need to do the research and development effort. So while it's very encouraging that we're that far along with Katahdins, we still have some background work we have to do on these other breeds. And we're going to need some participation from both our researchers as well as our industry flocks. And then there's a massive industry education need on the capabilities and implementation of this. We've learned an awful lot on the NSIP side and KHSI just over the last several months on how this is all going to work together, what the results are going to be, and how we can utilize that. We need to get that word out to the industry, which is what we're starting that process here tonight. But there's going to be a little bit of a learning curve as we get into this. So we need to figure out what the most efficient way is. So this current project, as we've mentioned several times, this was a project that KHSI, I believe the NSIP committee of Katahdin Hair Sheep International, submitted to the National Sheep Industry Improvement Center, and had several tiers to this project. One of the first tasks to overcome was to establish a DNA repository and that conduit for samples. So we're going through that right now as we're working through just that. How do we get the samples from the producers into NSIP in an efficient way into the evaluation? And then also, how do we store these DNA samples for use at a later date? Okay. Having these massive amounts of DNA available will help us down the road as the technologies evolve and as our research needs change. We can go back and access these historic samples so that we can keep building on the evaluation. As we said, we need an industry-wide informational program, and that's going to be multi-tiered. That's just on the general genomics information through this webinar series. There's going to be several different pamphlets and documents and fact sheets. Some of those are already in the works and already being developed. We'll be doing a lot more of those. Another key is developing sampling strategies. we know that, I mean, there's a cost to have these animals genotyped. Thankfully, it is much cheaper to genotype a sheep than it is a bull through the current strategies that we have worked out. But we still need to figure out, we know that there are animals that will contribute more information to the genetic evaluation than others. Okay, so how can we identify those animals? How can we strategically sample these animals so that we are getting as much usable information as possible, but doing it in a cost-effective manner? And then we'll also hoping to do some additional genotyping so that we can keep fresh current information going into the genetic evaluation. So this webinar series, since you all are here, you've all seen this flyer. I was just here to try to touch on this topic from a 30,000-foot view for what we can expect as producers. I highly encourage you to tune in and listen to these other three industry experts here, Dr. Ron Lewis, Dr. Scott Greiner, and Lynn Farmeyer and Tom Hodgman. All great resources that are going to help to elaborate on what I just kind of briefly touched on here this evening. If you want to know the kind of scientific details, I would definitely tune in to Dr. Lewis and Dr. Greiner's presentations here in the next coming weeks. So just to wrap things up a little bit, excuse me. Hopefully you were able to see kind of through these examples and through my lovely analogy of the back wall of my office, is that the whole point of what we're trying to do is utilize as much information as we possibly can so that we can make as an educated decision as possible when we're making genetic selection decisions. Turning all of this information that we're gathering into relevant decision-making tools that you all can use on your farms and ranches day in and day out. And that these tools, they can then be used to improve production efficiency, profitability, and sustainability. Because again, in today's modern agriculture, we need to be as efficient as we possibly can. So we need to use as much information as we possibly can so that our sheep flocks can be performing for us and be profitable into the future. So with that, I left a couple minutes for some questions. So I will gladly open it up. Any questions out there? Feel free to unmute yourselves or type them into the chat box. I didn't get a chance to look at the chat box much. I'll try to go through that right now. Mr. Hodgman, did you see anything big that came up that I wasn't able to get to?
We, between Michelle Campfield and myself, we did answer a couple of questions along the way. One originally was about each of the webinars being recorded, and those are going to be posted on with links to the recordings on nsip.org. That's I believe that's going to be under the genomic under resources / genomic enhanced breeding values. So that's a good place to look for that if you want to go back. And actually, I need to go back and check a couple of things that Rusty said. I wanted to be sure I understood what he was saying. So that's something I'm going to be doing. There was another question about Evvs. Do you know, someone asked if, is this similar to the Quebec-based program Evvs? And can you just briefly follow up on?
I know they do have they have a provincial system for the quantitative genetic evaluation. I don't know what work they are doing or have done on the molecular side, on the genomics, but I can certainly look into that. But
yeah, I was curious about it too. I was just poking around on their website, and it doesn't look like they mention anything about DNA or genomics. So they're just doing EBVs like we are doing in NSIP today.
There was a question, Lynn Farmeyer answered it, regarding the sampler, the applicator to be used to collect the DNA sample. The lab that NSIP is going to be contracting with will only accept Allflex, the TSU system from Allflex, which is widely available on on the web at this point, from what I've seen. So, I mean, just to follow through on that, no hair samples, no blood samples, just these TSUs collected in the device from the company Allflex is what will be accepted.
I saw there was a question there, kind of.
Go ahead.
What are the differences between some of the genotyping platforms? I'm sure a lot of us have heard about Flock 54, Zoetis. I don't know if they have a sheep platform or not. So Ron Lewis will be able to explain some of this, and I'm sure Dr. Greiner will as well. Right now, the only way to get the genome enhanced estimated breeding values is to be utilizing this 50K platform through NSIP. Some of these other platforms, like I guess I'm most familiar with Flock 54, so I'll talk about them. It's a great tool. They're able to provide the parentage, those known genetic conditions, but without tying it to phenotypic information, those platforms are really going to be they're going to be able to accurately give you information on the traits that are controlled by one or very few genes. Okay, so like the things like scrapie codon, for instance, that's what, 3 different genes that they're that we're looking at there. So those platforms can do that without tying it to the phenotypic information. It's difficult to shed much more information on the production side. So things like the reproductive rate, number of lambs born, number of lambs weaned, those traits, they're going to be controlled by probably thousands of genes. So that's where we need to have this broader platform and tying it to production information to generate these genome enhanced EBVs. I'm not really familiar with a lot of the other platforms. I said maybe Dr. Lewis or Dr. Greiner can answer that more, but getting these genome enhanced EBVs, the only way to get that on this performance information is to tie it to phenotypic information and incorporating it into the evaluation through NSIP.
Rusty, there was a question about signing up for additional, the other 3 webinars. And my understanding, and I'm looking at it on my phone at the moment, that there are links to signing up for the other 3 webinars at nsip.org under resources, and then under seminars and presentations. So perhaps Michelle or someone else could post those links. Another person asked if those links could be posted.
Yeah, I did, but here, I'm going to do it again because it kind of got buried in the chat.
Okay. Yeah. Thank you. Another person, Mike Duff, has asked about the difference between Flock 54 and Zoetis genomics. Can you do that, Rusty?
Yeah, that's what I was just trying to explain a little bit about the different genotyping platforms. I'm really not familiar with Zoetis's product for sheep, but like I said, the Flock 54, it's great for what it does, the parentage and the known genetic conditions. I know they're doing some work on that. I'm excited to see what they come up with in their next iteration. Been working with them a little bit on how we kind of see all this kind of playing together. And we kind of see 2 different kind of platforms or availabilities. We know that we're going to be able to get a lot more information from the 50K chips that we're working with and incorporating that information into the genetic evaluation. So what we're kind of thinking right now is that for our seed stock animals, these stud animals, the breeding stock, that we'll probably get the most bang for our buck using the 50K platform, but maybe our commercial producers that kind of want to get a snapshot of where their flock currently is. They don't want to generate genome enhanced EBVs themselves. They can go to one of these other platforms like Flock 54 to kind of get a good overall view of what their flock is currently doing. But it's really a matter of the biggest benefit to the 50K that we're working with is that it's tied to the phenotypic information and going into the genetic evaluation to produce genome enhanced EBVs.
Thanks, Rusty. Do you, Jerry Sorensen's asking about the cost of analysis on the 50K chip.
Yeah, I don't, I see Lynn's on as well. That's not proprietary, is it? As you understand, Tom?
I don't.
I think it's time to go public with this.
Okay, it's gonna, the cost is gonna be right about $30 for the genotyping information. So that's to get the animal genotyped and then pass through the system into the genomic evaluation.
There's also a question from Lori Marion about once a breed has established a sufficient genetic database, can the data be used to identify sheep that are unregistered but part of the breed as well as parentage?
Okay. I'm sorry, could you repeat that a little bit? I was trying to follow that.
Yeah, I had to read it a second time myself. I believe it's asking if you can use it to identify what breed or what breeds what breeds a sheep belongs to, what type of sheep it is. Once a breed has established a sufficient genetic database, can the data be used to identify sheep that are unregistered but part of the breed as well as its parentage?
Ooh, that's a great question that I'm going to have to pass along to Dr. Lewis. I'm sure the technology can do that, but in terms of estimating the genetic merit and being used for genome-enhanced EBVs, that I don't know if that would shed a whole lot of information for what we're trying to do. But that's something I'm going to have to I'll have to ask Dr. Lewis about. I mean, I know there are some breeds of livestock that are utilizing genomic tools to verify parentage to be used for registration purposes, to make sure that the sire and dam actually are who you say they are for registration, but I don't know if they're actually looking at breed purity. That's a great question.
Rusty, Ron can definitely go farther into this, but one of the beauties of molecular technology is that it doesn't necessarily have the same arbitrary bounds that we set on breeds, and so sometimes we can find a marker, or genes in an animal that are superior regardless of what we call the breed to be, and as we all well know, there's a lot of overlap molecularly between some of these breeds, and some animals that are described as pure aren't necessarily that way. So I think that going forward, there's a lot of opportunity molecularly to kind of break down some of the walls that we arbitrarily put up that aren't necessarily pure or have been respected for a long time.
Yeah, thanks, Joe. That as you were talking about that, I was just thinking about some of the preliminary work that Dr. Lewis did with some of his colleagues at University of Nebraska-Lincoln, and he was looking at the relationship of different populations within a breed. And I think that's the more important part, is how related are those animals, even if it is across breeds. I think there was some, we can probably find examples of animals of two different breeds that are pretty similar related. We're not going to get into that conversation tonight, but it's more important using these molecular techniques to identify how related animals are, not so much what breed we call that particular animal.
Well, even if it's just relationships like between a Dorset and a Polypay, or a Suffolk and a Katahdin, where there's definitely been influence in the composite, that we would expect there to be some overlap and some relationship. The lines are a lot more blurred than we necessarily describe them in the textbook.
Yeah, thanks a lot, Joe. Appreciate that.
Rusty, I have another question for you. Will we be able to link the NSIP database? I'm sorry. Will we be able to link the NSIP data to databases available in other countries if those animals are used through AI in the pedigree? For example, with the British Texel Basco. I believe we discussed this in our meeting with us, the Australian counterparts.
In some breeds we can,
Yes,
but it's because we work solely with Sheep Genetics Australia for our genetic evaluation. Those animals would have to be in the evaluation in Australia. So some of our breeds we were talking about just the other day that like our Dorpers and our White Dorpers and our SAMs, our South African meat merinos, we have sufficient genetic linkages between populations in Australia and populations in the United States is that we can kind of have cross references between those databases. In fact, the evaluation now for Dorpers and White Dorpers, US sheep are run right alongside the Australian sheep because we have such good genetic linkages. So if those linkages are there to the Australian populations, we can access those. But other countries outside of that, it might be a little bit difficult unless that information is in the system in Australia.
thank you. There's a question from Roxanne Newton about deciding on which lambs to collect from. Should we avoid collecting from lambs that have non-NSIP sires or grandsires?
Oh, that's another great question.
That's a very good question.
I would, I guess at this stage in the game, I'm not going to advise anyone not to collect samples. If that animal is going to shed some information within your flock that's going to be usable, I would encourage you to collect that sample if it's going to be economically beneficial for you. I do know that here in the very near future, there will be kind of a guidelines document of sampling strategies that Dr. Lewis has been working on with some of his colleagues. That will explain a lot of these strategies as to where we're going to get the most bang for our genotyping buck. So I'm going to pass the buck again to Dr. Lewis and let him give you a formal example.
There's a comment from Diane Almeida regarding, it's just a comment, it's not a question. In New Zealand, there are 3 genotyping companies that send the data to the New Zealand Genetic Evaluation, known as Sheep Improvement Limited. Through the single step of evaluation, breeders get GEBVs. I'll answer that. That's essentially the same concept that we're using now. Except it's one company doing the genotyping in the US, and the Australians will be doing the single step evaluation to get genomic enhanced breeding values for US producers.
Yeah, the Australians, they're working with 5 different genotyping companies, and NSIP, we're working with one currently right now, but it's not restricted to that. If there's other genotyping platforms that want to get that information into the evaluation, offer those services to the industry, we're open for it. We would just need to work with our colleagues in Australia to make sure that their platforms are accurate and they're getting the information that the Australians need for the evaluation.
Rusty, I have a good question for you. It's sort of an introductory question, but it's an important one, I think, especially for tonight's webinar. What if you don't have EBVs now? How will this process work, and how will it affect you?
That's a fantastic question. And it's easy and difficult to answer all at the same time. Essentially, if you don't have EBVs on your animals right now, you don't really have anything to compare all this DNA information to. Genotyping an animal is a fairly straightforward process. Just getting an estimate, or the 50K chip is going to look at 50,000 different places across that genome. But unless you use that, you compare that information to performance information, it's not going to tell you a whole lot. It's just going to tell you what the SNPs are at those 50,000 different places. So in order for these tools that we are talking about here today to be beneficial, is that we need to be generating EBVs on those sheep. Okay, so it's a fairly simple process to do that. You simply enroll in an NSIP and submit that data in for evaluation. But without those EBVs, without that phenotypic information, we can't generate genome enhanced EBVs.
Rusty, there's a question from a comment and a question from Dan Turner, and it relates to, he had heard a comment that genomic enhanced breeding values are similar to having X number of progeny in the system or X number of generations of data behind an animal. And he wanted to know if there was a common relationship such as this that we can use when we speak to others about the benefits of genomic enhanced breeding values.
Yeah, that's a great question, and I've heard it kind of presented that way too in other species. It gets a little bit difficult to say that having a 50K chip on an animal provides X number of, or the same data as X number of lambs, just because the benefits are going to be different on a trait by trait basis. Like I said, that for like our reproductive traits or low heritable traits, it's going to be tremendous value. So it's going to be the equivalent of, a larger number of lambs. But for some of those other traits like weaning weight, post-weaning weight, it's not going to help out very much on that. So it's kind of hard to give an exact number. Like I said, maybe one of the geneticists that'll be talking in the next couple of weeks can give you that number. But the typical extension answer of it is it depends.
And I'll follow up with a response from Lynn Farmeyer on that, back to Dan Turner, that where he also says it depends a lot on the trait and the number of relatives. But that's also a great question for Dr. Lewis. And as you know, this is the first of 4 webinars. So there are some of these questions that are clearly better designed to other speakers, but we're doing our best to get through them as informatively as we can. Mohammed Salma asked, he wants to know the differences between RAPD and ISSR techniques, which is used for breed evaluation, sounds like. Do you have any knowledge of that, Rusty?
I'm just trying to find that one. I'm not familiar with those abbreviations or that nomenclature.
Very near the bottom.
I Googled it because I was curious too. So RAPD is random amplified polymorphic DNA, and then ISSR is inner simple sequence repeat, and it's one of 3 different DNA-based techniques. So I don't know which one we're using or if we have any
These are sequencing techniques.
DNA nerds on the call who know the answer to that.
I'm going to no ifs, ands, or buts that one on to one of the geneticists. That is way beyond my reach.
So that's a great question, and thank you for that. I'm going to go to a question by Heidi Barber. Interesting you asked this question, Heidi. Are there training sites in each state to train producers that are looking to enroll in NSIP so they know how to accurately collect and record data? Rusty, you want to comment on that, especially after we spent some time last week going over some of this stuff? So it's interesting.
Sure. Yeah. So there's not like formal training sites in each state. There's a lot of different educational opportunities to get more information on NSIP. As Tom was just mentioning, a lot of us here on this call were just on a couple different weeks worth of webinar series that were hosted by University of Rhode Island. But if you're looking for more information, there's a lot of resources available out there. The NSIP website is a great resource, nsip.org. You can go to the resources tab under nsip.org to get more information about what data to collect, when to collect it. There's some standard operating procedures documents that kind of help to explain that. We also have a YouTube channel. We have a lot of different recorded webinars and tutorial videos that can help you with that. On the NSIP website, you can also find my contact information. So if you'd like to set up a training opportunity or a webinar or something like that, you shoot me an email and I'll be glad to work with you on it. Those are the big sources of information, nsip.org, and search National Sheep Improvement Program on YouTube. You can find that, find our channel there and see a lot of different videos that we have.
So I'm at the bottom of the chat, Rusty, and I just have Joe Emmenheiser's asked to pose a question to you before we wrap up.
Type that in there when it was a when it was a flurry, but it's slowed down. A really good job, and it's exciting to see this stuff taking shape. Just a kind of general breeds question. Taking a step back from the genomic enhanced EBVs for specific performance traits, I'm wondering if we could adopt this technology pretty quickly across NSIP, mostly just for not just parentage verification, but also, better quantifying the relationships between individuals. Right now we're just using pedigree expectations, and, we assume that all full SIBs, for example, share 50% of their genes, but that's not true on a molecular basis. Or quantifying molecular inbreeding, and we can, just with a basic 50K chip, get an inbreeding value that's better than our pedigree estimate, that can plug right into the same matrix and give us better estimates. And so I'm wondering if we would be able to get that technology in NSIP fairly quickly before we had the extensive test populations that you rightly said we need to have before we can have these traits in other breeds.
Yeah, that's a great question, Joe. And that's something that we've been working on quite a bit here is as we said earlier, that some of those initial benefits, as you mentioned, are simply getting an accurate pedigree and defining these relationships. So that was something we worked with literally just yesterday on our call with Australia, is that even though the genome enhanced EBVs won't be available for all of the breeds outside Katahdin, the parentage evaluation and the genetic conditions reporting, all of that can be taken into consideration for these other breeds. So you can send in DNA samples through that pipeline and get those parentage reports and those genetic conditions reports, even if you don't have genome enhanced EBVs yet. Another benefit to that is if we go through this conduit and go through Sheep Genetics Australia for that evaluation of parentage, when these breeds do have the sufficient research population established, those genotypes will be sitting in Australia ready to go in the evaluation when we turn that switch on. Okay? So doing some of that, we can do some of the background work now just through the parentage and genetic conditions, building up that resource population.
Mm-hmm.
Hey, Rusty, can I add a comment here?
Of course you can.
Yeah, Joe, I think as I heard your question, we need to ask the colleagues in Australia if their single step method will allow them to separate out the molecular question that you had before. I mean, you're asking kind of two things there. I think the single step method does the molecular work that you're asking for, but it also does everything else. And I'm not too sure we can separate out the phenotype and the need for the reference population with just the molecular parentage that you were asking about. But we could ask about that and then get back to you.
That would be good to know, because it sure seems to me that even before within a breed, and the test population and all those sorts of things, just a basic 50K SNP chip that's available to all breeds, we could use those identified SNPs, those 50K, to come up with an estimate of the percentage of those base pairs that two individuals have in common, and then that could be a substitute for the estimate that's used in the quantitative methods to make an EBV. So that it seems like that could be a preliminary step before all this other information is available that would go a long way to increasing accuracy, and to your point, be stored to help have a larger database when those production traits can come into the picture.
Yeah, I mean, I'm not a geneticist, but I understand what you're asking, and we'll run that question up the the chain of command, I guess, with the people in Australia and see what they say. I don't understand the single step method enough to know if that can be separated out, but we'll ask.
Cool, thank you.
Yeah, I'm sorry, I misunderstood that. I wasn't breaking out, I wasn't separating parentage from incorporating the genetic relationship into the evaluation. So yeah, as Lynn said, we'll bring that up with them and we'll get you an answer. But the parentage part of it, we know that the parentage part, that can be done.
Right, and we can do that privately without EBVs. Just send DNA to a lab and get it verified.
Correct.
You know,
Correct.
right.
Rusty, I have another question from Mike Duff regarding the role of Genetics USA. He asks, when NSIP becomes a part of Sheep Genetics USA, what is envisioned for how this will work? And I'm assuming he means the genomic analysis.
Oh, yeah, that analysis. Yeah. Yes, that's a great question, Mike. I guess I would, terminology, it might be semantics, but I wouldn't say that NSIP is becoming a part of Sheep Genetics USA. It's a lot of us on the NSIP board, myself included, have been working with Sheep Genetics USA to get that initiative started, but we're not. NSIP is going to be its own standalone entity. It's not going to be a part of Sheep Genetics USA. We'll work with Sheep Genetics USA hand in hand to help build these tools. Hopefully they'll be a good resource for NSIP on the research and development side, branching out these other breeds, developing some of these new traits. But NSIP is still going to be its own self-sufficient organization that's we're going to keep charging forward.
Thanks, Russ. That exhausts questions. If anyone has any others, feel free to unmute and speak up. It's about 30 minutes, almost last 20 minutes past our past time, so we've lost a number of participants. But if you still have a question or two, feel free to fire away. Lots of comments coming from folks. Rusty saying thank you so much for the presentation, and they learned a tremendous amount from it. So,
and I'm just typing into the chat box here. If anyone has any questions, feel free to shoot me an email or give me a phone call. I'd be glad to help out however I can. Thanks to everyone who came here this evening, and I'll stick around here for a few more minutes if anyone has any questions. But if you don't, have a great evening.
Thank you, Rusty. Bye.
Rusty,
Will this? Oh,
sorry,
no, go ahead.
I was tired of typing. I'm just curious. We're newer into the Katahdin sheeps. We've only had them about 4 or 5 years, and we bought an NSIP ram this past fall. So if we get into testing the offspring, should we only test the ones that are from registered ewes?
So, for testing, do you mean generating EBVs or using these genomic
The
tools?
genomics.
Yeah, so the genomics tools, I mean, it's anything that you collect and submit data on for evaluation through NSIP, you can utilize these genomic tools for. And that's not confined to just registered animals. So animals don't have to be registered to be a part of NSIP. So it's any animals that you'd like to generate EBVs on, any ones that you'd like to use these genomic tools on, and for genome enhanced EBVs, you'll be welcome to submit those samples as long as they are purebred Katahdins, if they're registered or not. Again, I would tune in for Dr. Lewis's. He'll help to shed more light on which animals it'll be worthwhile to be selecting for these genotyping platforms. I don't want you to mishear me and say I'm telling you to go and genotype every single animal in your flock. But if you'd like to, that option is available.
No, thanks. I can't afford that.
No, and as a new producer, with some animals that probably don't have a lot of links across the NSIP database, establishing those things is going to go 95% of the way to the accuracy, and the genomics really only add that extra margin. So animals like if you have 2 full sibling ram lambs that their estimated breeding values are pretty equal, but you do some molecular testing to see, well, this one really has more of the genes that have emerged as favorable for these traits. That's when it can be advantageous early on. But to test your whole flock is probably not particularly valuable right now. Ron can speak to that a lot more detail.
Thanks, guys.
But early on, establishing those genetic relationships, I think will be pretty big. So, but again, I'm gonna punt to the smart ones.
I would just mention, Candy, that there's some written guidelines coming out on that topic, but geared towards breeds that do not have a reference population currently developed, and then to Katahdins that do have a reference population developed. Just give you an idea of what you might want to consider in terms of who to do that, who to sample from, and that's being worked out with Ron Lewis right now. So look for those at nsip.org under Genomic Enhanced Evvs. You should be able to find that. It's not developed yet, although it's coming within the next month or so. All right, are we good?
Oh, Robert, I don't know if you got your question answered. Yes, we will. We will post this. It'll be up. We'll put it on the NSIP YouTube channel for sure, and I'll link to it in the NSIP website. I don't know. Is KHSI planning on posting it somewhere as well?
I'm sure we will.
Like I said, I'm glad to put it on the NSIP YouTube channel, and anybody who wants to link to it can. But I just got a text from Andrew Weaver, just landed. He said he'll get me the video as soon as it finishes.
Hey, Rusty, good luck hanging that picture.
Yeah. Thank you. I appreciate that. I appreciate that.
I hate to break it to you, but it's not going to be centered.
I don't think it will either. I don't think it will. I was trying to go through it though. I don't have a stud finder, so I had to Google a general picture of a stud finder because I'm too stubborn to go out and buy one. I just do the old knock on the wall trick and it works every time. But that didn't fit into the narrative I was trying to get across.
Well, thanks, Rusty. Appreciate you taking the time to do that for us.
Yeah.
Hey,
yeah.
you got another question in the
Oh, look
chat.
at that.
Speak about to differentiate between sheep breeds and how we can use genomic information to select good rams. Well, Mohammed, to utilize SNPs between different sheep breeds, the technology is going to be the same regardless of breed. If you're asking, can we differentiate one breed from another through it? Again, that's going to be something that Dr. Lewis will have to answer. I'm sure the technology is available, but it's not something that we're putting a lot of resources to in NSIP. But the technology is going to be the same. We can hopefully utilize these genomic tools to increase the accuracy of our quantitative genetic evaluation through NSIP. Having more accurate estimated breeding values will provide producers with the ability to utilize those EBVs in making selection decisions. So we can use those to select what we would consider to be our good rams from those that aren't. So it's across breeds, the technology is going to be all the same. It's just a matter of getting them set up within those particular breeds. So I don't think it's, if you're looking to say, should I be selecting a Katahdin or a Dorper? As of right now, I don't think the technology is going to be able to help you with that. But within that breed, it can help you to figure out which ones are the better ones. We have some other tools. We are trying to work on some across breed evaluations on the quantitative EBV side, but those tools they're not available yet. But hopefully we'll be able to utilize those to look at differences across different breeds.
Thank you very much. Thank you.
Yeah.
Thank you.
Did that answer the question you were trying to ask? I want to make sure I was understanding the question correctly.
Yeah, I understand it. Yeah.
Okay.
Yeah, thank you.
But I think this is a conversation for another day, but as we're trying to work towards these across breed evaluations, I would think that these molecular tools can also be used for that as well. But that's something I'm going to have to, does that make sense? Tom, Lynn, Joe? I know Ron's looking at the crossbred data sets from Sheep Experiment Station. I would think that if we have molecular information on the two pure, on those crossbreds, on those purebreds, that we should be able to do something across the two breeds, but.
Yeah, crossbreeding.
Yeah, well, we need to separate the crossbred animals from the crossbred evaluation. So when on crossbreds, I think of an F1 from breed A, breed B producing an F1 crossbred. Differentiating that though from comparing breed A to breed B without that F1 crossbred animal. Does that make sense? Because those are two different things.
Two different things.
Yeah.
That's part of what I was getting at, Rusty, though, with molecular inbreeding levels. We assume that an F1 crossbred has 100% heterosis, but the reality is that there's often overlap between those breeds, and even two parent breeds that we think are distinct, aren't. And so that 100% heterosis expectation in the F1 could be 90%, or it could be 95, or whatever the case may be. And that plugs right into the relationship matrix when these EBVs are calculated, where an individual that's inbred, or that's not, rather, has a relationship to itself of 1, but one that has a higher level of inbreeding has a relationship to itself greater than 1, which is a little counterintuitive, but that's how it works mathematically. And so being able to actually put molecular basis to those numbers it plugs right into the process that EBVs are already calculated by. So it seems like that would be a pretty easy step.
Yeah. I'm interested, since I raise polypays, if we still have the retained heterosis in that composite, because in a 4-breed cross, 25% each, we would expect 75% retained heterosis. I'd be interested to see, I guess to do that, we would have to have genotypes on all 4 purebred counterparts. So the fins might be the tricky one to have to get.
Right.
But yeah, I'd be interested to see how many generations we are from those original polypays, if that retained heterosis is still there, and how much inbreeding depression we have of that as well.
And it becomes particularly interesting when you get these groups or clusters within breeds, and the relationships between them. Like I'm reminded of Dave Notter's 3-way cross, which wasn't quite a polypay. It was a polypay with an extra shot of Dorset, but Dorset fin Rambouillet, and there were some that had higher numbers for aseasonality, and those were the ones that physically looked more Dorset. There were some that had high numbers for number of lambs born or weaned, and those phenotypically looked more thin. And then there were some that had higher growth numbers, and those phenotypically looked more Rambouillet. And that phenotype comes from the genes in some ways. And so I'm sure if those were genotyped, that there would be identified clusters within that composite breed. And you start breeding, the more Rambouillet phenotype, higher growth numbers to the higher prolificacy fin appearing, you're going to get a higher heterosis response than you will, from two within a cluster. So that stuff gets really cool when you get into these composite breeds.
Yeah, we definitely see that. I mean, I see that in my own polypays. It's the polypays that I have that are high for number of lambs born, number of lambs weaned. By visual appraisal, they look more like a fin. They're the finer featured, bald headed, and my ones that are more Rambouillet or Targhee appearing, those are my less prolific animals, higher growth type animals. So.
Yeah.
Rusty, I'm going to sign off. I have to check the lambing barn. So thank you very much. And I guess if everybody wants to keep chatting, go right ahead, but I have to go check on some ewes. So thanks again, and I'll sign off.
Thanks, Tom. Take care.
Yeah, thank you.
Yeah, I'm glad to stick around and answer a few more questions. As best I can as a nutritionist, not a geneticist. But my thought is if I can understand this as a nutritionist, just about anybody should be able to understand it.
Rusty, this is Mike Duff. Can you hear me all right?
Yeah, Mike. How are you tonight?
Wet. We're not bitter cold, but we've been getting rain and snow mix on top of everything. And we feed the ewes at night, so they'll lamb in the daytime, but we haven't started yet. So I'm out here this time of night slapping feed out, and so I'm wet, but I've been pleased to listen because I want to develop genomic enhanced EBVs for my flock, and I've tried some of these other platforms, and all I got was, of course, a comparison within my flock genetically, because their reference samples aren't big enough. I've submitted some reference samples to one program to try and get put in with some of my superior performers and whatnot. But how do I, and this is a $64 question for me for many years, how do I develop these EBVs with my Suffolk and Suffolk Camp cross genetics that are complete outcrosses from the NSIP, terminal sire and Suffolk groups? The gene pool is completely disconnected. And so how do I get a number that's meaningful? I have my own indexes now that I use and whatnot, but how do I get an EBV that can be useful to somebody across the country or across the world that wants to get an idea on my sheep?
Yeah, that's a good question, Mike. And that's the genetic relationship is kind of limiting there. If without genetic linkages to the other flocks in NSIP, the data is still only going to be comparable within your flock. These genomic tools might find some genetic relationship to some of the existing populations. It might not. I don't know. That'd be a good question for Ron Lewis. But the easiest way is to establish that genetic relationship somehow. It's swapping rams with somebody, or it's with your genetics. I'm trying to think if you would have any genetic ties. You're pretty isolated from the Midwestern flocks of Suffolks, right?
Yeah, there's very few, and there's been some cross-pollination via different other seed stock producers that have sold to me in the past, and I've used some of their genetics and some other people from the Midwest, but most of that stuff isn't in NSIP. I can think of one that might be, but it's not enough, you know. And I got 260 ewes, and we're growing to 360, and by keeping all our own genetics. But so the data that I can generate, and the data that I do generate already, it's mind-numbing to me. But the data that I generate there's a lot, but I'm starting to get progeny data from my customers because we've done the Block 54, we've done some genetic tests, and you know, Superior can kill my customers' lambs and test these lambs and send them back. And I'm wondering, is there an opportunity like there was in cattle at the beginning to develop some EBVs from using progeny test data? Because you know, right now I'm selling rams to large commercial flocks that are, you know, 3/4, 7/8, 15, 16 siblings. And so the genetic spread, you know, in the lambs, if I sell a guy, you know, I sold 30 to California this morning on the phone, and they're going to be 7/8 brothers. So all the lambs that are coming out of those ewes would, you know, conceivably have a pretty tight genomics pattern, given the vagaries we've already discussed in this presentation today. But and so I'm just wondering if there isn't a feasible way to take some of these outcross flocks that don't want to cross-pollinate genetically with the smaller Midwestern flocks, so that we can begin developing some EBVs that are meaningful. Does that make any sense, or am I on the ether cloud? I don't know, but
Yeah, no, I mean, that makes sense. That makes sense. It's all about trying to establish a genetic relationship somehow so that we can compare apples to apples. You're not the only one that's in that boat. I mean, there's a lot of producers that don't want to, you know, they don't like the existing gene pool for an NSIP for one reason or another, and that's perfectly fine. But we need to figure out how can we figure out these genetic relationships? Can these molecular techniques be used for that? That's a great question. I got to ask Ron that question. I don't know those capabilities. The easy solution that comes to mind for me is that I have had at least one person do this. Is that they were a terminal sire breeder. They didn't like the type of suffix that they had seen from NSIP. What they did is they went and they bought a Suffolk ram from an NSIP producer. They bred 10 ewes to them and then called all the progeny out of that ram just to establish that genetic connection. So they didn't keep anything out of them. They sold that ram right after the first year. They established their genetic connection, didn't influence their genetic lines long term, but they established their connection that they needed so they can be compared to the remaining suffix in NSIP. So,
right.
That's the simple, easy solution that comes to my mind without getting really complex with these DNA technologies. But like I said, Ron can answer that a heck of a lot better than I could.
Yeah. So if I bred, you know, 20 head of ewes and then compared those lambs to all the rest of my lambs, then that would be sufficient data to develop an EBV for the rest of my non-NSIP sired lambs. Is that what you're saying?
Well, if you collect data on all those other non-NSIP sired lambs, because then what that's doing is that's comparing these proven NSIP genetics it's proving the lambs out of that ram, comparing them to all the lambs that are out of your sires in the same contemporary group. So say, you know, you got 100 of your ewes bred to your rams, you've got 20 bred to this other NSIP ram, all lambing at the same time, one contemporary group. They're in a head-to-head comparison. You can generate EBVs on your entire flock that way.
Okay.
You know, that first year, yeah, you'll get EBVs for all the growth traits, and if you do carcass scans on carcass the first year, that first year might be a little bit difficult to have accuracies on the repro traits. But
well, that gives me an avenue to pursue, because you know, I would collect the data on all my other lambs, because I do, and you know, it's hard for you to make our flock. They got to really perform, you know, and so and same thing with the rams, but so I would do that, and I guess I'll
trying
have
to think, would you have any connections? I mean, do you have any lines? Do you work with Kitsons at all? Do you have any ties there? Or John Eagle would be another one. Would you have any genetic ties to either of them?
I don't have any genetic ties to John Eagle, but there may still be a thread or two with Kitsons. You know, I serve with Gwen on the American Lamb Board now, so I can talk to her about it. But they had some genetics coming from a couple of seed stock producers that I've bought rams from in the past, and whatnot. And how much of that they've retained, I don't know. But I know years ago they had some. But you know, other than that, you know, Matt. Now I'm having a senior moment. From Kansas, Nebraska, or
Beals.
Matt Beals. Yes, thank you for bailing out an old senile old fart. Matt Beals, because I talked to him on the phone last week. I'm sitting here going, what the hell? Anyway, I know he's on the hunt for a buck from a guy that's got some strong ties into my genetics too. So, but see, right now I've got 6 studs that I use, and only one of them was born off this place, and so and our line breeding coefficient is going to be off the charts. But if I could tie to Matt, or if I could tie to Kitson's, maybe there's a place to go there.
Yeah. Something we had done a couple of years ago is, I think it was Matt Beals that we had to do this for. Jeremy Geske was involved in it too, I think. But we assigned the Suffolk Association an NSIP ID number, so that we can utilize that in historic pedigrees, so we can establish these genetic linkages. So if you're indirectly connected to an NSIP flock, we can use that NSIP ID number for that third party or that missing link to help link up those pedigrees.
Okay.
So I know we did it for Matt Beals. I think we did it for Jeremy Geske as well. It worked. I mean, it's further back in a pedigree, or the more removed it is, the less impact that genetic relationship has on the evaluation, but it still helps to set up that genetic connectivity.
Yeah. And for me, you know, it's like the last time I did flock 54, it was page after page after page after page of the same damn thing. Because, you know, with all my rams, I tested every yearling ram, and this last year, and they were a hellacious crop of, they were performed, they were great, and you know, and that's how I paid $15 a head to get the flock 54 on damn near 200 grams, and the same data on the last page was on the first page, you know, and in between it was mind-numbing to just flip the pages and go, the number of the sheep changed, but none of the other data did, you know, it was all the same. And
Yeah.
and of course they couldn't do what I really wanted, which was give me the enhanced growth curve stuff. And
Right.
and I still I'm still looking for that, but any way I can get it. But
Yeah, you know, I mean, you and we can talk offline, but I think there's some solutions that we can find those genetic connections there, or you know, we can figure out a way to make that happen without, so that it's beneficial for the evaluation, but you know, you can use it to impact your breeding as much as you want. So I think there's some options there.
Okay, good. Let's talk, because I need to do it, and because it's like I say, it's getting really hard for my ewe lambs to make it into my flock using the system that I'm using now, and I want to make sure that there's no hole in my system. I'm letting good ones go down the road, and or the best ones, and then I want to make sure then that it's meaningful, that, you know, if somebody drove out here from Kentucky looking for a Suffolk buck, then they could have some EVVs that mean something to them. So
yeah, certainly.
But yeah, that gives me some hope. And but let me, one more question on this. If I got a buck, it would probably be smart for me to get the best NSIP buck I could get my grubby mitts on, wouldn't it? And compare mine to that buck, because from our phenotypic data, we're going to compare very well to high indexing NSIP sheep that I've seen sold in the last couple of years. And so is it better to get one of the top end bucks and try and compare the best that's there to what I've got, or to go average and compare?
I mean, it shouldn't matter. I mean, it's going to compare your animals to that line, regardless of if it's a good one or a bad one.
Okay.
Mike, if I can jump in, the very best move for you to do is to find a buck, regardless of what he looks like, regardless of what his numbers are, that's as well connected as any buck that you can find. If you can find an old buck that's been used in 4 different NSIP flocks, that's ideal, because that gives you that much stronger of a connection when you bring him into yours.
Okay. Well, like I say, if I was thinking maybe of leasing one and just sitting here listening to what you guys are advising me. If I leased one, got him in here, used him, and whatnot, we could get that jump start that we needed. But my prejudice is towards trying to find the best one somebody will let me borrow, but or rent, and go from there. Because I hate to say it, but there are traits that my customers pay me a lot of money for that NSIP doesn't measure, and I want to keep those without having to sacrifice them just to get numbers, you know. So, and I'm a contrarian. You know, it's like the scrapey thing. I didn't cull all my QQUs, you know, and everything, even though I'm export certified scrapey free and all that. I didn't cull a QQ sheep because I didn't want to discriminate against good sheep for a disease they didn't have. And because we're isolated, close flock and stuff. So I kind of think outside the box. Most people think I'm strange, but if I could do this the simplest way possible, but I'm willing to do it the hard way through collecting thousands and thousands of, sets of progeny test data to, because a lot of my customers retain ownership through slaughter. So they get paid on the pricing grid, so I can get that data back, and it would be helpful. But if there's an easier way, a faster way, then I'll try it. But Rusty can coach me on this, I guess.
Yeah.
And
I can help you on the basic side. Ron will have to answer you on the molecular, on the DNA side. I don't want to tell you that it'll work and have you go spend $30 a head for it to be a lie. So I can help you on the basic side. So there's probably some genetic linkages there that we can probably find right now just through a third party. And there's some ways that kind of like the situation that Joe was talking about, a well-connected buck. If you can find one that, like if you and Dwight could go in together on a buck and both of use them at the same, in the same year, would really help out that as well. So,
Okay.
Matt Beals has
There's
brought in a
options
couple
there.
bucks with that in mind too. He's just tried to find the best connected that he could.
Yeah. And
yeah.
and I hope he's supposed to be coming out here to buy a buck that is tied in with my flock, and I hope he brings a big enough checkbook and buys him so that then I can pirate data from Matt, but because it's going to take a big checkbook to get the ram. But I talked to him last week about it, and he's going to go for it. But so I hope that works. But if I could, I have hesitations about bringing outside sheep on my place. There's never been a sheep that came on my place in 32 years that ever left alive that I wasn't selling myself. And so, that's how we keep our disease-free status and all kinds of other stuff. And that, so I'd have to do some biological security protocols to make sure Ram's clean and everything when he gets here. But we'll try and work it out. Rusty, I'll let you coach me.
That's the easier way to do it, Mike, is convince Matt to buy one of your bucks, especially if it's one that you've already used. You can get the genetic relationship the same way by having Matt record them in his flock.
Well, I've got one he's interested in, and but I haven't gene tested him. I haven't done the flock 54 on him or even a gene check. So I'm going to do that this week, and I'm supposed to send some more samples to Brenda Murdoch for their sample pool and all that sort of stuff, and whatnot. But so I'll do that this week, and if Matt's still interested in him, we'll see. But there's a lot of people that like I say, think higher priorities on some traits than I do. So we'll see what they want and what he says when he comes to visit. But anyway,
I think we got options. You and I let's talk in the next couple of weeks, and we can get it figured out.
Okay, I appreciate that, and I appreciate you letting me ramble on here. But I mull these things over in my mind constantly while I'm socially distancing, and wonder how I can do this. And it's been my hesitation to be full bore into this thing, into NSIP for the last 4 or 5 years, was just having vertebrae enough to stand up and say, okay, we're going to bite the bullet and take this buck we really don't like and use him. But forgive me, but that's kind of where we've been at. And talking myself into doing it is one thing, talking my wife into doing it is another. So,
yeah.
But anyway, but I appreciate you letting me ramble on, and I'll get off and let you guys do what you're going to do and the rest of the night. But this has been helpful to me, and I've already signed up for the other sessions and stuff, so I hope to learn a little bit more and whatnot there. So.
Good. Well, good. So, well, thanks for coming, Mike. Appreciate it.
Well, thank you guys. Have a good night.
You too. Cody, like a blister, showing up when all the work's done.
Thanks a lot for calling me out. I missed this. I joined late, thanks to Joe Emmenheiser. If Mike is still on, I was about to send him a message saying that I appreciated his willingness, it looks like he's off, to think about NSIP, because he and I talked about it 2 years ago. So, and he was not considering it at all, but I liked what I heard. I heard that last conversation. That was about it. So
I've been working with Mike for 4 or 5 years now, just trying to get him to bite the bullet. He had signed up when we had the first year free deal and never participated.
Yep.
So we've been just trying to work through this connectivity issue, and maybe he's ready to pull the trigger.
I'm going to try to do my
know
left
it's not-
shoulder to the same as your right shoulder here.
Rusty, the line of the night was get an NSIP to buy your buck. I wanted to
Yeah.
virtually shake your hand on that one. Good job.
Yeah, I mean, I was about to write Mike and say, yes, exactly what Rusty said. Like, you don't have to buy a buck, but if somebody's interested in yours, you have that connection. Absolutely.
And I
So,
think he's got connections there. He just doesn't realize it. I know he's got connections to Kitson's. If he does the University of Idaho, they've got their flock on now.
mhm,
So there's some there that he just might not know about.
right. Yeah, to me, it was him just trusting the program. And the fact that he was here tonight listening, he trusts the program. So, or he's at least intrigued enough to look into trusting the program. So, and understanding that he can select for different traits that he wants to focus on that are different than, and again, same as last week, Joe's here again, looking at different traits that maybe some of the other Suffolk breeders are not looking to. You can still gauge your sheep on different traits rather than the norm of your breed. Is that fair?
I think that's fair.
So, I mean, I see there's just a few of us left here, and I don't, like Todd's here and a couple others, so I feel I can speak frankly. Joining in late.
I've been talking frankly for the last hour, so that's
That's good.
I
I miss these. Go ahead. Sorry.
I don't like I sugarcoat things, but it's I wanted to be open and honest with Mike. I mean, that's just, yeah.
I mean, that's all any of us are trying to be, is how do we breed better sheep? And let's talk about that.
Yeah. Yep.
So, yeah, thanks for calling me out for being the blister or pimple or whatever you called me. But yeah, I joined late.
Well, I thought since you joined us, how you wanted to contribute to the
I wasn't looking
conversation,
to it until you called
give
me
you the
out.
opportunity. Does anybody else have any other questions before we pull the plug? Once again, we got off on tangents and rambled for an extra hour, but
Damn,
Thanks,
all right.
Rusty. You did a great job tonight.
Yeah, it was really good. Just to clarify, all the different webinars in the series are a separate link, right? They're not, if we registered for this one, we're not automatically registered for the rest.
Correct. Yeah, I think you need
Okay.
to register separately for them.
Yeah.
Yeah. But yeah, all
Thanks,
those
Joe, for letting me know.
links should be on there.
Okay.
All right. Well, you guys have a great night.
Yeah. Try to stay warm.
We're going to try. I think one more day and we'll be hopefully ahead of it. So.
Yep.